Hi, good afternoon, everyone. Thank you for joining us for today's webinar. I'm Cassie, Marketing Supervisor at GenScript, and I'll be the moderator for the webinar today. A little background about GenScript for those who are new to us. GenScript is the leading life sciences research and application service and product provider in various fields, from basic life science research to translational biomedical development, industrial synthetic products, and cell therapeutic solutions. The topic for today's webinar is Emerging Trends and Technologies in Gene and Cell Therapy, and we have our scientific advisor, Dr. Leong Hao Wing, to present for today. Dr. Leong graduated from the National University of Singapore in 2011, majoring in pharmacology and neuroscience, and worked as a research fellow in Duke NUS Medical School, Singapore. She has more than 10 years of research experience in molecular biology techniques and extensive knowledge in protein structure modeling, protein expression, and its purification. Using these platforms, she deciphered novel cleavage sites of proteases and their functional implications in stroke upon cleavage of the ionotropic glutamic receptor. This webinar will take around 45 minutes and we'll have about 15 minutes for a Q&A session. You can type in your questions in the chat box and we will answer them after the presentation. If we didn't get to answer your questions during the Q&A session, we will email the answers back to you after this webinar. Do also help us to fill out the short survey that you can see on your screen and stand a chance to win a GenScript gift pack. So now, without further ado, we'll let Dr. Leong to start her presentation now. Dr. Leong, over to you.
Yep, hello. Thank you for the kind introduction. Good afternoon, everyone here. Thanks for attending this today's webinar. I'm Hao Wing, and I'm the scientific advisor from GenScript. It is my pleasure here to share with you the current trend in the industry on the emerging trends and technologies in gene cell therapy. So briefly for today's discussion, I will first talk about the current status of gene and cell therapy and its development. Then I will discuss some of the limitations in its R&D discovery and also the production of these therapies. Finally, I will shed some light on how such processes can be streamlined through some of the GenScript's one-stop solutions. So for the past 2 decades, the development of gene and cell therapy has been quite fast-tracked. And to date, there are already 22 FDA-approved GCT since the first approval in 2010. And among the 22 approved treatments, only two gene therapies are approved, namely Luxturna, to treat an inherited retinal disease, and Zolgensma, that targets the genetic root cause of spinal muscular atrophy, SMA, through the introduction of a functional gene, SMN1 gene. So the number of gene therapies approved are due to the complexities of the targeted disease for gene therapies, and also the lack of technologies and sometimes standards to efficiently deliver such gene treatment, which I will elaborate further in my later slides. So the rest of the approved GCT are cell therapies that involve various types of cell treatment. The majority of the treatment involves in the introduction of allogenic hematopoietic cells to treat patients with indications and disorder affecting the hematopoietic system. Such treatment derived from cord blood and produced in the bone marrow will be able to renew and differentiate into specialized cells. And when transplanted, these treatments are life-saving for people suffering from lymphoma, immune disorders, and also other related genetic conditions. However, in recent years, there have been increased interest in the CAR T-cell therapy. So within 4 years, from 2017 to 2021, there have been 5 first approved CAR T-cell therapy, and all 5 CAR T-cell therapies provide subsequent line of treatment for refractory or relapsed B-cell lymphoblastic leukemia. So it is not coincidental that the 5 CAR T cell therapy are approved within a 4 years time span, but rather through such much development and progress starting as early as in the 1957, when Barnes et al. Actually demonstrated in mice that the allogenic marrow transplantation successfully prevented a leukemia relapse, illustrating the activity of the engrafted immune system against the recipient. This is then followed by the work of Nobel laureate E. Donald Thomas, who first reported in 1957 the first allogenic hematopoietic stem cell treatment, HSCT, in patients, but resulted in the patients dying actually 100 days after. So through a better understanding of human leukocyte antigens HLA and advancing in methods in identifying and typing them, in 1979 he reported a cure rate of 50% using transplantation in acute myeloid leukemia, AML. And with such discoveries, new forms of cancer treatments evolved, with Steven Rosenberg and his colleagues in 1986 treating patients with tumor-infiltrating lymphocytes known as the TIL therapy. Such lymphocytes are removed from a tumor and expanded in the lab before giving back to the patients. So a few patients were cured of this advanced cancer from the treatment, showing that immune cells can fight cancer. And with a better understanding in the various types of cancer antigens, work on introducing gene into the T cells to engineer such immune cells as anti-cancer therapy was also started by Michael Sutherland in 1992, known as TCR therapy. And due to the limitations often found in TIL and TCR therapy, in terms of the number of T cells and the major histocompatibility complex, the MHC incompatibility, efforts have been made to overcome this via synthetic receptors. So the first chimeric antigen receptor CAR, the first generation, was actually a fusion of antibody fused to the T-cell receptor. And this was developed by Salik Esha in 1993. So with such a discovery, such immune cells, such as T cells, are either boosted or genetically altered to enhance its cancer fighting capabilities. And many refer this to gene and cell therapy, GCT, in which the T cell are engineered through knocking off the genes. For example, CAR, to eliminate cancer and other diseases. And this is one such aspect of the GCT, as mentioned in earlier slides, and could involve in also TIL, TCR therapy, CAR T-cell therapy, and also recently the introduction of CAR into natural killer cells, NK cells, as therapy. And usually in gene therapy, it involves in the replacement and the introduction, a replacement of a defective gene with a functional gene, as in the Lusterna, the treatment Lusterna. Genes can be introduced either in vivo, by directly introducing it into the human body, or ex vivo, where specific cells, for example, T cells, are first collected, and then the gene is engineered in vitro into the cells and expanded in culture before its reintroduction into patients in an autologous manner. So, but definitely there are some limitations, as mentioned, in terms of why is there such a low incidence of this kind of gene therapy. Such limitations include, that's, due to the essence of gene itself. So only 10% of the genome can be targeted by the conventional rationale, Lipinsky rule of 5, as small molecule drug design. So it leads to inefficient delivery of this of such nucleic acid to their correct site of action, and also poor toxicology profile in some viral-based factors. Example, due to adenoviral, for example, adenoviral stimulation of the immune response. There also need to be more cell-specific And it needs to penetrate, through the cell into the intracellular lumen. And more often than not, such genes, to be introduced back into the cells are usually of high molecular weight, and they are also vulnerable to enzymatic degradation. Another properties of this is that the genes are usually anionic, which makes them poor translocators to the cell membrane. And of course, as mentioned, it has to be more target specific. So currently, there are various ways of introducing such genes. The common one being the virus. And of course, there are also non-viral gene delivery. For non-viral gene delivery, it can be by, in vivo method, as mentioned earlier, through naked DNA transfer by electroporation. Or if it is in vitro, there are also several methods available, such as biophysical, such as electroporation, gene gun, microinjection, ultrasound, and hydrodynamic delivery. The common chemical ways of introducing such gene would be, through liposomes, polymers, dendrimers, and also sometimes peptides, which can aid in changing the properties of the gene, to allow them to be properly transferred through the cell membrane. So currently, the most common methods, as mentioned earlier on, it includes the foreign method besides the virus, such as the lentiviral factors, and also another one, which is the retroviral factors. For the non-viral methods, it will be, transposon and transposase system, and also mRNA transfer. Each has its own advantages and disadvantages. For example, one of the earliest method that was used, which is the retroviral factors, the advantages it will be being, it usually can ensure, high expression of the genes into the T cells, and it has been, it has a long history of being used for such transduction. So there's a relatively, high in vitro safety related to these retroviral factors. However, in terms of there are always, concerns about, safety concerns in vivo. And that's why there needs to be long-term monitoring of these patients receiving these retrovirally transduced T cells. And moreover, because of it being using viral vectors, there are complex manufacturing, expensive testing that needs to be included during its manufacturing to reduce the possibility of having replication competent virus. So for lentiviral factors, one of the advantages for it would be, is able to transduce non-dividing cells, and also at the same time, there is it is compared to the retroviral factors, it's similar to the retroviral, but that is better in that it is safer. And it has also a high efficiency and stable expression. However, there is the disadvantages would be the lack of a stable packaging system, and then there can be variability between the lots, and also similarly, because it being viral, there needs to be complex manufacturing and testing to ensure there is no replication competent virus. So that's why sometimes there will be alternatives approach using non-viral factors, such as the transposon and transposase systems, such as the piggyback and the Sleeping Beauty. The advantages, of course, will be, it is simpler to manufacture because they are non-viral, and of course that's why they are comparatively the manufacturing cost will be lower compared to the viral factors. for mRNA transfer, it is also good because there is no genomic integration and there is no risk of replication competent virus. However, the disadvantages of it being it is only a transient expression, so that is not really ideal. When cells are back into the patient itself when it is only a transient expression. So that's why, considering of all these pros and cons, the lentiviral factors are still, the preferred one of the more preferred method to transduce, genes such as CARs into the T cells. And currently, that as such, in order to reduce, the risk involved, there have been many improvements and developments to make such lentiviral factors as safe as possible. So the first generation used to be, a 3 plasmid system, whereby there are still some holds that kind of a HIV accessory components, which will increase the risk of generating this kind of replication competent lentiviruses. So subsequently, with the second generation, they have changed the way, that it is packaged. The TET, REF, and GET-PO genes are all on the same packaging plasmid. However, it is still driven by a TET dependent wild type promoter. As such, from there have been further development with the third generation and some improvements in terms of the LTR region. It is using a chimeric 5' UTR with a deletion of the U3 region of the 3' ETR at the same time. And also in terms of to have a better control, the REF is on the single plasmid with the TET removed, and GET-PO is on another plasmid. And as such, it can be TET independent and can be driven by other viral promoters such as CMV. And this it uses a 4 plasmid system. Yes, definitely the yield will be decreased, but it is a much safer alternative. So just in terms of upscaling, considerations have to be made to ensure the production can be of a high yield. So in GenScript, our lentiviruses are, because we understand many of these lentiviruses are to be used, for transduction in T cells. We have developed our lentiviruses such that, they can transduce T cells with a stable expression over time. They are able to handle hard to transduce cells as shown by the MOI. We only need very little amounts before, it can have a large number of positively transduced cells. It worked very well with T cells such as the Jacquard cells, and they are stable after prolonged passages, even until even until passage 30. And of course, as mentioned, that needs to, especially when such viruses are to be used, in terms in the transduction and the production of T cell therapy, such as CAR T cell therapy. It will be important to ensure the yield will be good in producing this lentivirus to reduce the cost. Currently, there are several methods of generating such lentivirus using usually the HEC 293 system. It can be in the adherent culture, cell culture, or the suspension culture. Each has its own characteristic and advantages. For example, in adherent culture, the characteristic is that it is anchorage dependent. There needs to be cell adhesion, and they are usually grown as a monolayers. And passaging will be required, and this will be detachment of the adherent cells from the surface, and plating on the new surface in a fresh medium will be required. One of the advantages of this adherent cell culture would be, it has been very much well developed with very mature methodology available. While the limitations would be to grow such adherent cell culture, most of the time, serum will be needed, and usually this may not be optimal for downstream purposes as regulatory purposes, as there will be concerns about adventitious virus, and care has to be taken to remove, possibility of such viruses as much as possible. And also at the same time, the growth is limited by surface area, and that's why it will have limitation on the yield, and it is also difficult to scale up. Another way of generating large scale the lentivirus would be in terms of suspension culture. In suspension cultures, these cells are actually floating in the medium, and passaging is done easily by diluting them into fresh medium. So it can ensure, less contamination when using suspension cell culture. So one of the advantages will also be it is easy to scale up, and because suspension cell culture then need not be can be done in serum free, thus decreasing the risk of contamination by adventitious agent. So the limitation is that sometimes, there will be higher impurity content in the supernatant, and care has to be done to improve on its clarification and also the filtration. So a comparison between, the adherent and the suspension, as mentioned in some of the earlier slides, would be, like for adherent cells, they will be on a single layer, then requires to be a substrate for attachment, requires a periodic passaging and detachment of the adherent cells from the surface, and that's why that may be issues with difficulty in scaling up. So in comparison, a suspension kind of culture, it does not require substrate for attachment, and they are easy to passage. It can be easily diluted into fresh medium, making them easy to scale up. And as such, in GenScript, we have considered such needs for large-scale production. That's why we have also developed our own proprietary 293 T cells. We call it the Power STM 293T to reduce the cost of production of these lentivirus. Compared to other commercial cell lines, the Power S 293T suspension cell line, are able to have very high LVV titer. For as shown here by 6 different gene of interest, 5 different gene of interest, and also it can have stable performance ranging from 30 mil to 50 mil. As shown by the titer in FACS, and also by the ELISA titer on P24. And because of our proprietary cell line, we have improved in terms of the yield in such culture compared to the adherent process. For example, it increases 10 times in terms of in the supernatant in terms of the titer that can be obtained compared to the adherent process. Because of this, there is actually about 40 to 60% reduction in cost per unit in terms of the production of this lentivirus compared to the adherent system using the Power S293T cell lines that we have in GenScript ProBio. So not only we understand the needs of using viral method to produce ways of transducing CARs or T-cell engineering, or this more traditional way of CAR introduction into the T-cell via viral vectors. GenScript also strive to overcome some of the concerns using viral vectors, such as the increased risk of insertional mutagenesis, and of course, as mentioned in previous slides, those high cost and long lead time for manufacturing. And also with viruses, and also some of the transposon-based approaches using a homology-directed repair, it can allow, precise, more precise targeting. Targeting the genomic changes. As such, it can improve the quality and the uniformity and the safety of the cellular products. And because of the reduce in the integration risk, it can also improve the insertion into a specific locus, such as, for example, for the CAR, the TRAC locus, which is the T-cell receptor alpha chain constant region. And such, because of this, it can increase the CAR T-cell potency and also its persistence. So this homology-directed repair, which is Cas-mediated knock-ins, are good for large payload, unlike currently available genomic editing tools such as a base editor or prime editor or transposases or recombinase. But of course, with such a homology-directed repair, there are some limitations. Usually, there need to be a higher level of DNA that is required compared to non-viral approaches because usually it is done through electroporation. So as such, there can be instances of DNA toxicity. Yeah, and sometimes an alternative method would be, still, the viral method to introduce this homology-directed repair. To improve, compared to electroporation. As such, it will be back to the same concern about, expensive manufacturing of viral factors to introduce such genes through the homology-directed repair. So as such, GenScript has, worked with Dr. Shai from the Marsden lab to produce a non-viral delivery of using CRISPR and Cas9 editing tools and payload template DNA to enable site-specific transgene insertion via the homology-directed repair HDR with minimized toxicity to reduce regulatory concern. So what we did was we developed a hybrid single-stranded DNA, ssDNA DRT with a Cas9 targeted sequences, ssCTS, to boost the knock-in efficiency by more than 5-fold and live cell yield by more than 7-fold. This takes the advantages of using the ssCTS low toxicity at high concentration. However, the challenge is the production of long ssDNA sequences, and through collaboration with us in GenScript, we have developed these long ssDNA sequences, and now we're able to perform therapeutic gene editing in primary T cells. The hybrid ssDNA HDR template comprise of these small stretches of dsDNA incorporating the Cas9 targeting sites, CTS, and through a new complementary oligonucleotides, and in addition to the homology arms at each end of the gene insert. So the added CTS can then interact with the Cas9 ribonucleoproteins, RNPs, to shuttle the template to the nucleus and enhance, in this way, enhance the HDR efficiency without having the need to have high concentration of DNA. So in this figure, the development of these large CTS templates as shown for different gene knock-in across different target loci for high efficiency and low toxicity HDR in primary human T cells. For example, this TNGFR knock-in at the IL2RA locus with 78.5 knock-in efficiency, and this construct is about 1.5 kilobase construct. And we also tried it on the IL2RA GFP fusion protein knock-in at the IL2RA locus with 38% knock-in efficiency. And this construct is at least 2.3 kilobase And we also tried on the BCMA CAR construct at the track locus via two different gRNA, G526 and also G527 with a knock-in efficiency of 39%. And this construct is at least 2.9 kilobase. And the use of this SSCTS with large ssDNA constructs outperform other templates as shown here, compared to just the ssDNA and also the dsDNA with higher knock-in efficiency and lower toxicity. And that includes the dsDNA CTS. So we also evaluated the performance of across several different primary cell types such as CD4 plus T cells, CD8 plus regulatory T cells, TRAC, NK cells, B cells, and also hematopoietic stem cells. Using a mCherry knock-in construct in this instance. And for we are confident with our ssDNA to produce precise and non-toxic and efficient knock-in as well as stable dsDNA for large gene knock-in. So for our ssDNA, we can offer up to 5 kilobase and mid-scale count production. And also our ssDNA are all sequence verified. We can do both reverse and forward primer sequencing, and only the desired strand will be used to show up with key results and high purity. We also offer stable dsDNA production with a covalently close ends to minimize NHEJ risk and also to increase better insertion accuracy. Our dsDNA can go up to 10 kilobase and in gram scale and can be more affordable compared to the ssDNA. So besides, the limitations of introducing genes into the T cells, one of the other major limiting factors would be the production of these engineered T cells. Some of the concerns will be, whether there will be a proper activation of the T cells, whether, there will be such T cells will have the ability to maintain its activity for enough time to sustain the effective anti-tumor response. Also, whether there will be enough number of T cells that can be collected, for this kind of T cell production. At the same time, there are also concerns about the tumor heterogeneity. As some of these engineered T cell, they are very specific towards certain cancer antigen. However, in real life cases, tumor are very heterogeneous. Whether we could be able to use such therapy to target, the heterogeneous tumor population. Also, there are also concerns with such CAR T cell therapy that, many instances, therapies are done in this immunosuppressive tumor microenvironment. As such, whether such T cell are actually optimal, it is in that optimal environment to effect its anti-tumor response. And also because of this autologous T cell generation, it is quite restraining for large-scale clinical use, and it is on a case-by-case basis also because of its nature of autologous T cell generation. And also, as mentioned, it can be costly, in terms of producing such a T cell therapy, not only with the lentiviral production, but also, the gene engineering and also the need to have GMP levels or a very tightly regulated kind of production to minimize, the kind of a possibility for contamination, for example. And currently, the CAR T cell therapy is mainly for lymphoblastic leukemia, and there is still a lack of solution for solid tumors. And most importantly, the specificity towards the antigen, towards the cancer, has to be very specific to reduce the side effects that can come with such a T cell therapy. So in view of all the different limitation, GenScript had also worked on to provide solutions. For example, one of the proprietary technology that we have, in isolating this T cells, the CD4 plus and also the CD8 plus cells would be using the cytosine bits that we have developed proprietarily. So it is based on the immunomagnetic system. It is biodegradable, making them non-toxic. And using the stably conjugated antibody, it is able to efficiently pull down CD4 plus or CD8 plus cell. So as shown here, we have very high purity after using these bits for purification, as shown by here, the CD4 plus population, the purity, and also the CD8 plus purity, with high recovery rates for both cell types, and they are very viable. As shown here in the graph, there's minimal differences after the cell separation, before and after isolation. And also these cells, after isolation and after the separation using the beads, they are readily able to be activated, as shown here, the CD4 plus cells and also the CD8 plus cells, and they are readily expandable. And compared to some of the competitive beads out there, our beads are also better, such that we are able to be very specific towards our cell population, as shown by our CD4 plus positive cells with very low amount of monocytes and NK cells, and also CD8 plus cells and B cells, compared to the competitor cells, competitor beads. And also this is for the CD8, the results for the CD8 plus, as shown here. So not only so, as mentioned, currently we need to have more efforts and development to overcome, limitations such as it being more for to be more for solid tumors, and also that's compatibility or improvements in the tumor environment that can for such a CAR T-cell therapy. As such, besides as early as the first generation, usually it was the first generation was usually was actually via this single chain scFv antibody attached to a CD3 via the hinge region and the transmembrane region. This, however, such a CAR are not will not be able to allow persistent T-cell activation and survival. And as such, a co-stimulatory domain such as the CD28 that will allow the interaction with CD8T and CD86 ligand on target cell surface will be included in the second generation. And the third generation and actually include different co-stimulatory domain to improve the functionality and also the tumor responses, and also to overcome some of the issues being it's more for to improve the possibility of such CAR in solid tumors, and also improve its functionality in the immunosuppressive environment. We have there have been trends to develop the fourth generation of the CAR, or we call it the truck CAR or the armored CAR, whereby the NFAT can be included to stimulate and boost the CAR T in the anti-cancer capabilities through the expression of cytokines such as IL-12 or antibody-like proteins. So in addition to, having, this fourth generation of CARs, development of this fourth generation of CARs to, open up the possibility of CARs in solid tumors, and also to improve its function in those immunosuppressive environment often seen in such a solid tumor environment. In addition, there have been efforts to improve, the specificity and also the binding strength towards the target through the use of VHH. So the VHH is only harboring the heavy variable chain, and these are usually typically produced by the camelid. And using the VHH, such VHH have a higher solubility and higher stability, and because it allows for dimerization and multimerization, as such it can increase the binding strength and specificity towards the antigen itself. So these are some of the current development, of CARs to improve the CAR T-cell function. So in view of that, in GenScript, we also would like to help researchers in streamlining such CAR development. So GenScript has a series of services for CAR discovery and also production, such as we are able to do a VHH discovery, a CAR-led discovery. Let's say if we produce the antibody already, we can also do it in an in vitro manner. We have the precision mutational library, which can actually improve the affinity maturation of this kind of antibodies in vitro to obtain the best antibody or best scFv or VHH or nanobodies for your CAR T-cell development. We can also provide developability assessment, affinity measurements, and many a times this introduction of this lentivirus or the gene itself requires GMP manufacturing. We also provide such GMP manufacturing of lentivirus and also plasmid DNA. We also provide cell line development for clients who are interested in looking into the efficacy of the CAR T, and we also provide cell banking. For the CAR-led discovery, we have several options, such as using the hybridoma. We also are equipped with a single B-cell screening on a chip to increase the discovery process, the number of antibodies that can be obtained. We also provide, the SDAB, the VHH production, and we have both the immunized library and also the naive library available for such discovery. So we are able to provide scFv or VHH domain candidates. We can provide, affinity ranking such that you can choose the most optimized targets for your CAR T cell therapy generation. We can include humanization of such antibodies. If further affinity maturation is required, as mentioned, we have the in vitro method, which is the precision mutational library, which is the in vitro method to actually improve on this affinity maturation of these antibodies. At the same time, we will be able to provide, the various kind of CAR T construct generation. We will also be able to do the functional bioassay, for example, cytotoxicity assay, or the related cytokine release or proliferation assay. At the end, we'll provide deliverables of 1 to 3 good sequences for this CAR-led discovery. So as shown here, the lentivirus produced can allow very good transduction of our CARs, and these are some of the killing assay that we have done for this CD19 CAR T cells, and its effectiveness of a lysis on the Raji cells. And this is one of the cytokine release IFN gamma that we have done with this CD19 CAR T cells. So as mentioned, not only we have, you know, the mice and the camelid platform available, we also have the rabbit platform available. In essence, you know, sometimes, you know, the rabbit, because of its several rounds of affinity maturation in that immune system, it can produce, you know, higher specificity, higher affinity kind of antibodies. And we have in GenScript also developed the B-cell cloning approach to increase the number of positive clones that could be obtained using the rabbit immunization approach. So briefly, you know, in this B-cell cloning approach that we have, the animal will be immunized with the respective antigen. The PBMC will be isolated. The antigen positive B cell will be enriched and expanded, followed by its screening and the harvesting of the supernatant. There is no requirement of the fusion with myeloma, such that we can increase and ensure a large cell pool for our Western blot or FAT screening. Up to 10 to the power of 5 memory B cells can be seeded and screened. And at the end, you will obtain, you know, the antibody sequence, and the sequence and the IP itself belongs to you. Yeah. And because of the lack of fusion, it can be as fast as the antibodies production can be as fast as 13 weeks. In addition, we to development of CAR, you know, a production of antibodies, be it scFv, VHH, or maybe isolation of the T cells, we understand, you know, many a times, you know, the target itself is important, and identification of these targets are never easy. So there are different types of cancer antigen, for example, tumor-associated antigen, whereby usually they are processed fragments of proteins, normally in low expression in other cells, but high expression in cancer cells. Some good examples and currently used are like the CD19 for B-cell malignancies. And we also have the cancer of testis CT antigens, usually present in reproductive tissue and not in adult tissue. And they appear only in a subset of malignant tumors, for example, the MAGE2 and also the IMYSO1. Of course, there are also the viral antigen, which is usually associated with viral infection. And usually the cancer cells expressing the proteins, they encode this viral open reading frame. Example of this would be the human papillovirus or the cervical oropharyngeal cancers. And of course, the neoantigen, which is a mutation in the protein coding region in non-germline cell after birth. And these are somatic mutations that can accumulate in cancer cells during the initiation and development and the metastasis of the tumors. So there are many ways in identifying this kind of cancer-specific antigen. One way which would be the whole cell genome sequencing. Another way would be through in silico peptide prediction based on the HLA binding affinity or proteasome processing to identify such peptides. And of course, another method would be the LC-MS based kind of immunopeptidomics to identify such a new antigen. So in GenScript, we recognize, you know, that the needs of such identification of this cancer-associated peptide. As such, we have also formed a collaboration with Cayman to perform immunopeptidome profiling, which is a deep sequencing analysis of the MHC-associated peptide to identify such neoantigen or cancer biomarkers. There are immunogenic sequences from MHC1 or 2 So briefly, the process is the human or the mouse tumor cell are lies under optimal conditions to release the intact MHC complex, and then using immunoaffinity chromatography, the MHC specific antibodies will enrich the pool of MHC class 1 or 2 from the lysates, and these peptides are then released under gentle eluting conditions and separated from the captured MHC molecules. And then the peptides are then sequenced by LC-MS and then matched through protein sequence database or even RNA sequencing data, as well as analyzed for mutation and also post-translational modification to facilitate the identification of this potential new antigen. And in GenScript, we have lots of experience in producing such difficult, many times, hydrophobic new antigens. So as shown here by T and O in molecular cellular proteomics, this is one of the ways whereby they use a combination method to identify a new antigen. Not only with the MHC immunopeptidome profiling, but also on the whole exome genome sequencing, and then they compared with the databases and also from the de novo search, and then they identify several different kind of potential peptides, and these peptides are then synthesized with GenScript, for them to test, whether they are indeed potential new antigen targets. So not only we have, the aforementioned some of the services that we have to for GCT R&D and also production. We have actually been around for 19 years, supporting 600K projects with about nearly 40% working in GCT R&D. We have at least 3,500 GCT publications that are using GenScript reagents. And some of the services we can provide not only like the ssDNA or CRISPR service or viral packaging or antibody discovery, peptide synthesis. We are known for our molecular biology to produce good quality kind of plasmid DNA, GMP grade also, and also protein expression. As such, we have, not only the aforementioned services, we also have different kind of solution for GCT, like the mutant libraries to target for engineering, CRISPR services, and also stable cell line for lead identification. Okay, I hope that I have all this information put together. I hope that I have shared some of the important or emerging trends and technologies in gene and cell therapy. Thank you so much for your time.
Thank you, Dr. Leong, for the great presentation. We have a few questions from the floor right now, so we'll answer it one by one. So the first question is, can you provide the reason for the preferential expression of CAR construct in CD8 2B cells over the CD4 2B cells when CRISPR-Cas9 approach is used?
That's because of the actually it is also being able to use on the CD8 cells. Yeah. So currently both cell types are also but with the CD8 cells being the more preferred one for that kind of cytotoxic kind of activity. Yeah.
All right. Thanks, Dr. Leong. The next question. How effective is GenScript antibody magnetic beads compared to mutiny?
We have done, we are not able to share, which are the competitors that we have tested on, but definitely with the, as shown by the results, compared to the competitor, which is one of them, which will be the gold standard of the mutiny, we do have comparable or even better kind of isolation in terms of the CD4 plus and also the CD8 plus. If interested, you can always get in touch with us, and we will be able to maybe even provide some free samples for you all to test out and compare on your end, and then test it out on yourself with yourself by yourself. Yep.
All right.
Yeah.
Thanks,
Just
Dr.
contact
Leong.
us for more information. Yep.
Yeah, that's right. If you need, we can send you a free samples to test it out. So the next question, how do you conjugate peptide to DNA?
So sometimes, as mentioned, there are several different kind of methods whereby we can use to introduce DNA. One of them would be, the peptide. For example, if it is, I have not delve too much in it, but usually they will be able to form the polylysine homopolymers can be used to condense with the plasmid using the calcium chloride, or it can be done using a biotinylated form of the peptide, and then it can be linked via a avidin moiety. Yeah.
Okay, thank you, Dr. Leong. The next question, what can I do if I have a library with too many hydrophobic residues?
Oh yeah, yeah. Sometimes for neoantigen, there can be issues with them being hydrophobic. So sometimes they may be difficult, for its synthesis, difficult to purify and also solubilize. So in GenScript, we have a peptide calculator. If need, I can share with you the websites that we have that can probe into the characteristic of the peptides to have a better understanding of the properties and the peptides and how easy it is to synthesize. So if the libraries have many hydrophobic peptides, we can either choose lower purity or substituting some non-crucial hydrophobic residues to others, such as glutamic acid or lysine, or other peptide sequences can also be used if possible. Yeah.
Okay, thanks, Dr. Leong. The next question, why does rabbit has a superior diversity of antibody generation compared to rodents?
Oh, okay. I have briefly mentioned, there are several rounds of enzymatic maturation, or more precisely, somatic recombination and also somatic hypermutation at the VDJ region. And for the rabbits, usually it happens in the gut, and so it is the gut-associated lymphoid tissue, which is very similar to the mammal in essence. So this neonatal immature B cell development and early postnatal primary B cell development has already been exposed, the gut microflora, and that's why they are able to result in stronger secondary affinity antibody maturation processes. And in addition to that, rabbit also goes through 2 rounds of this SHM, one during the mature B cell development and also one during the adaptive immune response. So because of this, and also because of the heterogeneity and the evolutionary distance from humans, they can compared to the mice, right? They will be able to recognize a wider range of these immunogens. So that's why, in essence, compared to the rodents, sometimes in terms of producing high affinity and specificity kind of antibodies, rabbit is superior compared to the rodents.
Thank you, Dr. Leong. We will answer the last question from Desai again. What is more preferable, CD4 or CD8 CARs?
Usually CD8 CARs are more preferable due to the cytotoxic. But actually, there has also been, a trend of having, NK cells, because of its innate mechanism, to have the cytotoxic properties. Yeah.
Okay, thank you, Dr. Leong. So we will end the Q&A session over here. So let's say if any of you have any further questions, you can just write into us and I will email back all the answers from Dr. Leong back to you. So once again, thank you, Dr. Leong, for the great presentation.